Welcome to the deep dive. Today we're going to be looking at how extrachromosomal DNAs are inherited in cancer cells.
hmm
There was a really fascinating paper in Nature recently about it, and you sent this in, so you obviously know the background on medicine and cancer already. So we're going to skip all the basics and just get right into how these ecDNAs all work together and how they're passed on when the cell divides.
Well, the paper really focused on these ecDNAs, which are circular DNA units. They're not part of the chromosomes. They're actually floating around outside of them. And they're known to amplify oncogenes, which is one of the things that really fuels tumor growth and heterogeneity. And one of the interesting things that they found in this paper is that by analyzing data from the Cancer Genome Atlas, they saw that over 25% of tumors that have ecDNA actually have multiple distinct ecDNA species coexisting within a single tumor.
Wow.
And that's really fascinating because it kind of goes against the grain of what we understand about traditional inheritance, where genes on the same chromosome are typically inherited together. So it raises a whole bunch of questions about how these different ecDNAs are interacting and being passed down through generations.
Right. Yeah, that makes sense. So how did they actually go about looking at this? Like, what kind of specific things were they looking for?
So they picked 5 cancer cell lines to study. They looked at COLO 320DM, which is a colorectal cancer cell line, SNU 16-MILO1 for gastric cancer, TR14 for neuroblastoma, and then GBM 39KT and CA 718 for glioblastoma. And they picked these because they were known to have multiple ecDNA species.
Yeah.
And they confirmed that these distinct ecDNAs weren't just, linked together on the same molecule. They were actually existing as independent units within the same cell.
Oh, wow. So they weren't all just like part of one big chain.
Right, exactly. They used a couple of techniques to do this. They used DNA fluorescence in situ hybridization, or FIH as it's known. And then they also did some single-cell sequencing to confirm that these were actually separate units.
That's really cool. Okay, so they've got these cells. They've confirmed that these ecDNAs are all separate. What do they do next?
Well, the next thing they did is they analyzed the copy numbers of these distinct ecDNA species within single cells. So they used a bunch of different techniques for this. They used FESH imaging again. They use SCATAC-SEC, which is a way to look at accessible chromatin regions in single cells. And they use CIRCLE-SEC, which specifically focuses on circular DNA in single cells.
Wow. So they were really looking at it from all these different angles. That's really neat.
Yeah, and what they found was actually really surprising. They found that there was a really strong correlation in copy numbers between different ecDNA species.
What do you mean by that?
So basically, if a cell had a high copy number of one type of ecDNA, it tended to also have high copy numbers of other types of ecDNA.
Oh, interesting. So it wasn't just random. Like if one was high, the others were more likely to be high as well.
Exactly. And this correlation was really consistent. They saw it across all of the cancer cell lines they studied, and they also saw it in patient tumor samples.
Wow. So this wasn't just some fluke in the lab. This was actually happening in real tumors.
Yeah, and this was a pretty unexpected finding because it really suggests that the inheritance of ecDNAs might be more coordinated than we previously thought.
So it's not just like they're all randomly being passed on. There's actually some kind of system going on.
Right. And to figure out what was going on, they decided to explore 3 possible explanations.
Okay, maybe those.
So the first explanation was hyperreplication.
Hyperreplication. What is that?
Basically, the idea that some cells might just be overproducing all types of ecDNAs, just making way more copies of everything.
Okay. That makes sense. I guess that they're just making more of everything than you'd see more of everything.
Exactly. But they pretty quickly ruled this out as the main factor. They did some experiments, and it just didn't seem like hyperreplication was the primary explanation.
Okay. So what were the other 2 explanations?
The other 2 were co-selection and co-segregation.
Okay. So co-selection, I guess that means that certain combinations of ecDNAs give a survival advantage. So they're selected together.
Exactly. That could explain why different ecDNA species tend to end up in the same cells.
Okay. But that doesn't really explain why their copy numbers are so closely linked, right?
Right. Co-selection doesn't fully explain the tight correlation in copy numbers that they saw within individual cells.
Right, because co-selection just means that they're both there.
Uh-huh.
It doesn't mean that there's necessarily the same amount of each one.
Exactly. And that's where co-segregation comes in.
Okay, so what is co-segregation?
Co-segregation is basically the idea that different ecDNA species are passed on together to daughter cells during cell division.
Oh, so it's not just random like we thought. Like they're actually being deliberately kept together.
That's what it seems like. And to actually visualize this, they did a really clever experiment. They combined AVEA with immunofluorescence.
Immunofluorescence. That sounds complicated.
It's not that complicated. Basically, it's a technique that uses antibodies to visualize specific proteins.
Oh, so they could actually see the ecDNAs and the proteins that are involved in cell division.
Exactly. And this allowed them to track the distribution of the ecDNAs during mitosis, which is the process of cell division.
So they could see where the ecDNAs went when the cell split in two.
Right. And what they saw really challenged the idea of random ecDNA inheritance.
What did they see?
They found that daughter cells that inherited more copies of one ecDNA species were significantly more likely to also inherit more copies of the other species.
Oh, wow. So it wasn't random at all? It was like the ecDNAs were sticking together.
Exactly. And this co-segregation pattern was something they saw consistently across all 5 of the cancer cell lines they studied.
Wow. So it's like ecDNAs have some way of knowing to stick together and make sure they both get passed on.
That's what it looks like. And that was a really big deal because it really changes the way we think about ecDNA inheritance.
Yeah, it's not just this random process. It's something much more organized and coordinated.
Right. And that raises a whole bunch of new questions, like what's driving this co-segregation? How are these ecDNAs sticking together?
Yeah. What's the mechanism behind all of this?
Well, that's what they wanted to find out. So they started by looking at the potential role of DNA damage-induced clustering.
DNA damage-induced clustering. What is that?
Basically, it's the idea that when DNA gets damaged, it can sometimes cluster together in certain areas of the cell.
Okay, so they thought maybe the ecDNAs are being clustered together because of DNA damage.
Right, that was one possibility. So they did some experiments where they knocked out the CIP2A gene.
CIP2A, what does that gene do?
CIP2A is a gene that's involved in clustering damaged DNA fragments.
Oh, so if they knocked out that gene, then they could see if DNA damage was really the cause of the clustering.
Exactly. And what they found was that knocking out CIP2A didn't actually affect ecDNA co-segregation that much.
So DNA damage wasn't the main driver.
Right. It seemed like something else was going on.
Okay. So what else did they look at?
Well, they then turned their attention to the role of transcription.
Transcription, and it's a process of making RNA copies from DNA, right?
Exactly. And they used live cell imaging to see what was happening. And what they observed was that ecDNAs often cluster together in these transcriptionally active hubs.
So they're all hanging out together in places where the DNA is being actively transcribed.
Right. And what's really interesting is that these hubs persisted throughout mitosis.
So even when the cell was dividing, the ecDNAs were still clustered together in these transcription hubs.
Exactly. And that made them think that maybe these hubs were playing a role in co-segregation.
Okay, so how did they test that?
They used a drug called triptolide.
Triptolide, what does that do?
Triptolide is a drug that specifically inhibits the initiation stage of transcription.
So it stops the process of making RNA copies from DNA.
Right, and what they found was that when they inhibited transcription with triptolide, it actually reduced ecDNA co-segregation.
Wow, so inhibiting transcription was actually preventing the ecDNAs from sticking together.
Exactly, and that really suggested that active transcription, particularly at those initial stages, plays a really crucial role in orchestrating ecDNA inheritance.
That's fascinating. So it's like the ecDNAs are using the process of transcription to coordinate their movement.
That's what it seems like. And they actually did another experiment to confirm this. They used nascent RNA Phe.
Nascent RNA Phe. What is that?
It's a way to visualize newly synthesized RNA molecules.
Oh, so they could actually see where the RNA was being made?
Right. And what they found was that, unlike chromosomes, ecDNAs actually remain transcriptionally active at the beginning of mitosis.
So even when the cell is starting to divide, the ecDNAs are still making RNA.
Exactly. And that further supports the idea that transcription is playing a key role in ecDNA co-segregation.
Wow. So it's like the ecDNAs are using transcription as a way to signal to each other and make sure they're all moving together.
That's one way to think about it. And this was a really big finding because it really changes the way we think about how ecDNAs are regulated and how they behave during cell division.
Yeah, it's like they're not just these passive pieces of DNA. They're actually actively participating in the process of inheritance.
Right. And that has really big implications for how we understand cancer development and how we might be able to target ecDNAs for treatment.
That's really interesting. So they've got this idea of co-selection and co-segregation. How did they figure out how much each of those was contributing to what they were seeing?
Well, to do that, they actually developed a computational model.
A computational model. That sounds pretty high-tech.
It was pretty cool. Basically, it allowed them to simulate ecDNA dynamics in a growing tumor population.
So they could create a virtual tumor and see how the ecDNAs behaved over time.
Exactly. And this allowed them to explore the effects of co-selection and co-segregation on ecDNA co-occurrence and copy number correlation.
So they could see how much each of those factors was contributing to the patterns they were seeing in the real tumors.
Right. And the simulations actually revealed some really interesting things.
Like what?
Well, they found that co-selection is the primary driver of ecDNA co-occurrence.
So that means that different ecDNA species are ending up in the same cells because they provide a combined survival advantage. Okay, but what about the correlation in copy numbers?
Well, that's where co-segregation comes in. The simulations showed that co-segregation is the main reason for the observed correlation in copy numbers within individual cells.
So co-selection gets different ecDNA species into the same cell, but co-segregation keeps their copy numbers coordinated.
Exactly. And this was a really important finding because it showed that both co-selection and co-segregation are playing a role in shaping ecDNA dynamics.
That's really cool. So they've got this model that can explain how these ecDNAs are behaving. What else did they do with it?
Well, the model is actually really useful because it can be used to infer the levels of co-selection and co-segregation from experimental data.
So they can take data from real tumors and use the model to figure out how much co-selection and co-segregation is happening.
Exactly. And that gives them valuable insights into tumor evolution and the intricate dynamics of ecDNA inheritance.
That's really powerful. So they've got this model that can help them understand how these ecDNAs are evolving and spreading within a tumor. What do they do next?
Well, they didn't stop there. They actually went on to explore the concept of ecDNA specialization.
ecDNA specialization. What does that mean?
Well, they found a really intriguing example of this in the SNU16 gastric cancer cell line.
Okay. What do they find?
They found an enhancer-only ecDNA.
An enhancer-only ecDNA. What's that?
Well, it's basically an ecDNA that doesn't actually contain any oncogenes itself.
No oncogenes. So what does it do?
Instead of oncogenes, it harbors enhancer sequences.
Enhancers. What are those?
Enhancers are regions of DNA that can regulate gene expression from a distance.
Oh, so they can turn genes on or off even if they're not right next to the gene?
Exactly. And what they found was that this enhancer-only ecDNA was interacting with the FGFR2 oncogene.
FGFR2. That's an oncogene, right?
Yeah, it's located on a separate ecDNA molecule.
So this enhancer-only ecDNA was actually boosting the expression of an oncogene on a different ecDNA.
Exactly. And that was a really significant finding because it showed that even ecDNAs that don't contain oncogenes themselves can still play a crucial role in tumor development.
Wow, so they're like little helpers. They're helping the oncogenes do their thing.
Right. And this really highlights the complex interplay that can happen between different ecDNA species.
So it's not just about the oncogenes themselves. It's about the whole network of ecDNAs and how they're interacting with each other.
Exactly. And to confirm that this enhancer-only ecDNA was really sticking with the oncogene, they used FISH analysis again.
And what did they find?
They found that the enhancer-only ecDNA was co-segregating with both the MYC and FGFR2 ecDNAs.
So it was being passed on together with the oncogene.
Exactly. And this coordinated inheritance is really important because it ensures that the enhancer stays associated with its target oncogene.
Right, so it can keep boosting its expression.
Exactly. And this really highlights the importance of co-segregation for maintaining stable oncogene cooperation.
That's fascinating. So it's like the ecDNAs have evolved this system to make sure that the oncogenes have all the help they need to do their thing.
That's a good way to put it. And this finding really challenges the traditional view of genetic inheritance.
Yeah, it's not just about the genes themselves. It's about how they're organized and how they're working together.
Right. And this is something that we really need to consider when we're thinking about how to treat cancer.
That's a great point. We need to understand the whole system, not just the individual parts.
Exactly. And that's one of the things that makes this research so exciting. It's opening up whole new avenues for understanding cancer and developing new therapies.
I completely agree. This is a really fascinating area of research, and I'm excited to see where it goes from here.
Me too. There's still so much to learn about ecDNAs and how they're shaping cancer development.
Well, I think this paper is a great step forward, and it's really given us a lot to think about.
I agree. And I think it highlights the importance of continuing to invest in basic research so we can keep uncovering these important insights.
Absolutely. Well, thank you so much for taking the time to explain all of this to us. It's been really enlightening.
My pleasure. It's always great to talk about this kind of cutting-edge research. So this research really shows that These ecDNAs, they're playing by a different set of rules than our normal chromosomes,
Mm.
right? They're adding this whole other layer of complexity to how cancer develops and how it evolves.
Yeah, and I guess that's what makes it so important to understand how these things work.
Hmm.
Because if we're trying to develop treatments for cancer, we need to know how these ecDNAs are behaving and how they're being passed on.
Absolutely. And one of the things that the study highlights is that if you target one ecDNA species, it can actually have an indirect effect on the copy numbers of other ecDNA species that are co-segregating with it.
Right, because they're all linked together in a way.
Exactly. And that could be a good thing if it means that you're depleting multiple oncogenes at the same time. But it also raises some concerns about resistance. If one ecDNA species manages to escape by integrating into a chromosome, it could continue to drive tumor growth. And it could even potentially contribute to the resurgence of those other ecDNAs that were co-segregating with it.
Oh, wow. So it's like they're helping each other out even if one of them gets taken out.
Right. And that really shows just how adaptable cancer cells can be. They're constantly evolving and finding ways to survive.
It's like a constant arms race.
Exactly. And that's why we need to have a better understanding of how ecDNAs behave so we can develop therapies that can effectively target them and prevent resistance from emerging.
Right, so it's not just about finding the right drug. It's about understanding the whole system and how all the pieces fit together.
Exactly. And another really interesting finding from this paper is the discovery of these enhancer-only ecDNAs.
Yeah, I was really intrigued by that. What exactly are those again?
So these are ecDNAs that don't actually contain any oncogenes themselves,
Yeah.
but they can still have a major impact on oncogene expression.
How do they do that?
They do it by providing enhancer elements.
Okay. And what do enhancers do again?
Enhancers are basically stretches of DNA that can regulate gene expression from a distance, so they can turn genes on or off even if they're not located right next to the gene. Oh, so it's like they're sending out signals to control the oncogenes.
Right. And what they found was that these enhancer-only ecDNAs were interacting with the FGFR2 oncogene, which is located on a different ecDNA molecule.
So even though they don't have any oncogenes themselves, they can still boost the expression of oncogenes on other ecDNAs.
Exactly. And that's really important because it challenges the traditional view that oncogenes are the main drivers of cancer.
Right. It's not just about the oncogenes. It's about the whole network of genetic elements and how they're interacting with each other.
And this discovery of enhancer-only ecDNAs really raises some interesting questions about how these specialized ecDNAs came about and how they evolved.
Yeah, like what's the evolutionary advantage of having these enhancer-only ecDNAs?
Right. And how do they maintain their association with those oncogenes over time?
Those are really great questions. And I guess that's something that researchers will be looking into in the future.
Absolutely. It's a really exciting area of research. And it could lead to some new insights into how ecDNAs evolve and how we might be able to target them for treatment. That's really cool. And the researchers also suggested that these principles of coordinated inheritance that they observed with ecDNAs might actually extend to other cellular elements as well.
Right. They think it might apply to things like viral episomes, subcellular organelles, and biomolecular condensates.
Wow. So it's not just limited to ecDNAs. It could be a much broader phenomena.
Exactly. And if that's the case, it could have major implications for our understanding of a whole range of biological processes, not just cancer.
That's amazing.
Yeah.
So this research could have implications far beyond what they initially set out to study.
Exactly. It's a great example of how basic research can lead to unexpected discoveries that can have a big impact on our understanding of biology.
And the researchers also developed a really cool computational model as part of this study.
Yeah, the model is really useful because it allows researchers to simulate ecDNA dynamics and explore the contributions of co-selection and co-segregation under different conditions.
Oh, so they can basically create a virtual tumor and see how the ecDNAs behave.
Right. And they can use the model to infer the levels of co-selection and co-segregation from experimental data. So it's a really valuable tool for understanding how these processes are shaping ecDNA inheritance patterns.
And I guess that could help researchers develop more effective treatments.
Absolutely. By understanding how ecDNAs are evolving and spreading within a tumor, researchers can start to develop therapies that can target them more effectively.
And the researchers also emphasized that their findings have some important implications for how we think about cancer treatment in general.
Right. They argue that we need to move away from this idea of just targeting individual oncogenes.
We need to start thinking about the tumor as a whole system.
Exactly. We need to understand how different ecDNA species are interacting with each other and with the cellular environment.
And we need to anticipate how those interactions might lead to resistance.
Right. And we need to develop therapies that can overcome those resistance mechanisms.
So it's a much more holistic approach to cancer treatment.
Exactly. And it's going to require a deeper understanding of the molecular mechanisms that are driving tumor growth and adaptation.
That's a really big challenge. But I think this research is a great step in the right direction.
I agree. It's really exciting to see these kinds of advances being made in the field of cancer research.
It gives me hope that we'll be able to develop more effective treatments in the future.
Me too. There's still a lot of work to be done, but I think we're moving in the right direction.
And I think this paper really highlights the importance of continuing to invest in basic research.
Absolutely. Because you never know what kind of amazing discoveries might be just around the corner.
It really highlights just how different these ecDNAs are from normal chromosomes.
Right.
They're not following the same rules of inheritance.
Exactly. It really adds this whole other level of complexity to understanding cancer.
And I think one of the biggest takeaways from this research is that we really need to start thinking about ecDNAs when we're developing cancer treatments.
Absolutely, because as they showed in the study, targeting one ecDNA species can have these unintended consequences on other ecDNA species that are co-segregating with it.
Right, because they're kind of all connected in a way.
Exactly. And that could be a good thing or a bad thing. It could be good if it means that you're knocking out multiple oncogenes at the same time, but it could also lead to resistance if one of those ecDNAs figures out a way to escape.
It's like a double-edged sword.
Exactly. And that just goes to show how adaptable cancer cells can be. They're constantly evolving and finding new ways to survive.
So it's not enough to just target one thing. You really need to understand the whole system.
Right. You need to think about the tumor as this complex ecosystem with all these different players interacting with each other.
And one of those interesting players in that ecosystem is these enhancer-only ecDNAs.
Yeah, those were a really surprising finding. It's amazing to think that these ecDNAs that don't even have any oncogenes themselves can still have such a big impact on tumor development.
It's like they're the puppet masters behind the scenes.
Right, they're pulling the strings and controlling the oncogenes.
And that really changes our understanding of how cancer works. It's not just about the oncogenes themselves. It's about a whole network of genetic elements that are working together to drive tumor growth.
Exactly. And this discovery of enhancer-only ecDNAs really opens up a whole new area of research. We need to figure out how these specialized ecDNAs came about and how they evolved.
Right, like what are the evolutionary pressures that led to their development? And how do they manage to stay associated with those oncogenes over time?
Those are really important questions, and I think the answers could lead to some really exciting new therapeutic targets.
It's like we're just scratching the surface of understanding ecDNAs.
Absolutely. And the more we learn about them, the better equipped we'll be to develop more effective cancer treatments.
And I think one of the most interesting things about this research is that it might have implications beyond cancer.
Right. The researchers suggested that these principles of coordinated inheritance that they observed with ecDNAs might actually apply to other cellular elements as well.
So it could be a much more widespread phenomenon.
Exactly. And that could have implications for our understanding of a whole range of biological processes, not just cancer.
It's really amazing to think about.
Yeah, it just shows how interconnected biology is and how discoveries in one area can have ripple effects across other fields.
Yeah, and the researchers also developed a really impressive computational model as part of this study.
Yeah, the model is really cool. It allows researchers to simulate ecDNA dynamics and explore the contributions of co-selection and co-segregation under different conditions.
So it's like a virtual laboratory where they can test out different scenarios.
Exactly. And it's a really powerful tool for understanding how these processes are shaping ecDNA inheritance patterns.
And I think that kind of modeling is going to become increasingly important in cancer research as we start to generate more and more data.
Absolutely. Because we need ways to make sense of all that data, and computational models can help us do that.
They can help us see the patterns and the connections that we might not otherwise be able to see.
Exactly. And they can help us develop more effective treatments by giving us a better understanding of how cancer works.
And I think the researchers made a really important point about the need to move away from this idea of targeting individual oncogenes.
Right. We need to start thinking about the tumor as a whole system and targeting the network of interactions that are driving tumor growth.
Because if we just focus on one thing, the cancer will find a way to adapt and escape.
Exactly. It's like playing whack-a-mole. You knock one thing down and another one pops up.
So we need to be smarter about how we approach cancer treatment.
We need to think about the long game and develop therapies that can target the whole system and prevent resistance from emerging.
And that's going to require a much deeper understanding of the molecular mechanisms that are driving tumor growth and adaptation.
Absolutely. It's a big challenge. But I think this research is a really promising step in the right direction.
I agree. It's really exciting to see this kind of progress being made in the field of cancer research.
Yeah, it gives me hope that we'll be able to develop more effective and less toxic treatments in the future.
Me too. I think we're on the cusp of a really exciting new era in cancer treatment.
I agree. And I think research like this is paving the way.
Well, this has been a truly fascinating deep dive into the world of extrachromosomal DNAs and their role in cancer.
It's amazing to think about all the complexity that's going on inside these cells.
And it's really inspiring to see how scientists are using cutting-edge techniques to unravel those complexities.
Absolutely. It's a testament to the power of human ingenuity and the importance of continuing to invest in scientific research.
Because who knows what other amazing discoveries are waiting to be made.
Exactly. The possibilities are endless.
Well, thank you so much for joining us on this journey into the fascinating world of ecDNAs.
It's been my pleasure.
And thank you to all of our listeners for tuning in. We hope you found this deep dive as informative and engaging as we did.
We'll see you next time on The Deep Dive.